Alright guys, good morning. What we're going to do today is we're going to go through the eukaryotic transcription stuff. I'm going to stop before we get to the translation stuff because I know some of you guys are having questions about like initiation, elongation, termination. That's probably going to be a separate video. So what we're going to do right now is we're just going to go through the eukaryotic transcription stuff because it's vastly more difficult than prokaryotic. Obviously, you've got a lot more DNA because you've got all these chromosomes. It's not just like one circular chromosome floating around a nucleoid region of a bacterium. So we've got a lot more stuff that we got to deal with. And because we have more stuff, that means we have more enzymes and they have different jobs. So if you remember, there are different types of RNA. The different types of RNA have different jobs. We have ribosomal RNA. That's where we're going to assemble our finished polypeptides or our proteins. We've got tRNA. tRNA is going to bring us the monomer subunits that we actually need to build our proteins. So that's going to bring us our amino acids and put them together. And then we need our mRNA because that's going to be essentially our recipe. It's telling us what to build, what amino acids do I need and what order do I need to put them together in. So we've got different polymerases for different jobs. All right, I'm mostly going to focus over here. Yes, there's some information over here, but primarily what you need to know lives over in this area. So RNA polymerase 1 is going to make the ribosomal RNA. That's its whole job. It goes ahead, it reads the code to make ribosomes. RNA polymerase 2, its job is to basically manufacture mRNA. It's going to basically open up your cookbook of DNA, read one recipe, and write it down so we can send that mRNA out of the nucleus, out into the cytoplasm, to the ribosomes to manufacture our final protein product. And then we've got RNA polymerase 3, that's going to manufacture our tRNA. Again, T stands for transfer. That's going to go ahead and bring us our amino acids that we're going to need. Each of these polymerases is going to have its own promoter on the DNA. So there's a certain area on the DNA where basically these things are going to be, the enzymes that are going to manufacture these are going to be looking for that area and going, okay, here's RNA Pol 1, here's RNA Pol 2, and here's RNA Pol 3. Okay? Alright, so like I said, that's pretty much what I need you to know there. Now, because it's eukaryotic transcription, we got a couple of different things going on here. Alright? Much like prokaryotic, okay, prokaryotic transcription had that sigma subunit where those two fingers were spaced just so. So they would find that -10 and -35 area upstream from where you actually start to write down or copy down your new DNA, or your mRNA rather. Same thing's going to happen here. So we have a transcription factor, and its first job is to basically look for this thing known as a TATA box. And it's called a TATA box because it's just a series of thymines and adenines. That's all it is. So it's a big long stretch of T's and A's and T's and A's. So this transcription factor basically looks for that, and you can see by its shape it's going to clamp down on that. Now, once this transcription factor clamps down on the DNA at the right spot, what happens is it starts to recruit all these other transcription factors. And these transcription factors are going to come, they're going to join in a certain way that you're going to get every time this exact kind of spacing. And that spacing from having these extra guys here is going to allow RNA polymerase 2 to sit down at a particular spot. So notice, from here to here, there's going to be a certain amount of space, and that's going to be basically our initiation point. That's where we want to start kind of reading the DNA. All right. So back here, this is kind of a precursor. It's like, hey, that's the title of the recipe that we're going to go ahead and copy. And here's where we actually start reading the recipe and copying it, essentially. Okay. Now, one of the things that we have to do after we go ahead and we make our mRNA, and that's all we're going to do. I mean, I can go back here, but once we start doing this, you're just going to make mRNA, which you guys know how to do transcription. So, you know, A's to U's, G's to C's, so on and so forth. Now, once we actually manufacture our stretch of mRNA, and you can see it's got a 5' end and a 3' end, we have to send the mRNA somewhere. The place it has to go is into the cytoplasm. The cytoplasm, there's a lot of different chemical reactions going on, and some of these reactions that are going on could damage our new mRNA transcript. And we don't want that to happen. That's our recipe. If we screw up our recipe, we're going to make the wrong product. We're going to make the wrong protein. If we make the wrong protein, it's not going to fold up right. It's not going to do its job correctly. So what we're going to do is we're going to protect it. And we're going to protect our mRNA, which is this yellow section here, and we're going to protect it with what they call a 5' methyl cap. Now, a methyl group is basically just a CH3. Okay, so we put this CH3 over here. It doesn't like to react with a whole lot. So essentially, this protects this beginning end of our mRNA. That's going to be like our start side where we're going to start to read the mRNA and manufacture our protein. Well, I also don't want to have the back end of my mRNA transcript messed with. There's information in there like stop codons and stuff, and if we broke one of those off early, we might get what's called a truncated protein. It'd be shorter and it wouldn't be functional. So back here, I take a bunch of adenines, whole bunch of adenines, and I just slap them on there. If those get damaged, it's okay, because in here, I'm probably still going to have my stop codon protected. So I put these protective caps on each end because there's lots of different reactions going on out in the cytoplasm. I know that was a question that some of you had about the poly-A tail and the methyl cap. Hopefully that satisfies that curiosity. I think it should. All right. Now, I'm sorry for this picture. I had to take it with my phone out of the textbook, and I know it's a little weird. But a lot of you guys are asking about splicing. All right. Now, here's the thing. Not all DNA is considered useful DNA. Some of it's just kind of these sequences that are kind of thrown in for conservation of DNA. And basically what's going on is we have exons. These are the sequences that are expressed. These are the sequences of genes that we're actually going to turn into a protein. And we have these intervening sequences, these sequences of junk DNA. It's not really junk, but we need to get rid of it. So as you can see here in this picture, here's our DNA template. And we can see we've got exons and we've got introns. The blue parts up here, the exons are what I would want, and the purple parts, I want to get rid of them eventually. That's my non-essential junk DNA. All right, so I'm going to go ahead, I'm going to read my DNA, I'm going to make my mRNA transcript, I'm going to put my 5' cap and my 3' tail on it, because I got to send my mRNA out to the cytoplasm so it can get turned into a protein. But before I want to turn it into a protein, we're going to do splicing. It's what they sometimes call post-transcriptional modification, right? So before I get into the whole translation, I'm going to take these pieces of mRNA that are non-essential, I'm going to get rid of them. So I'm going to cut them, and I'm going to splice the yellow pieces back together to get my mature mRNA. Now what this means is that every time you make a piece of mRNA, when it gets sent out to the cytoplasm, it's not necessarily ready to be turned directly into a protein. And depending on how you splice these things back together, you can get a lot of different proteins, okay? So, as far as the splicing reaction is concerned, what's going to happen, okay? The intron and the exon junctions, which remember this is going to be a series of A's and T's, and there might be little markers in there. They're going to be recognized by these small nuclear ribonuclear phosphates, if I remember correctly, RNPs, small nuclear ribonuclear protein particles. And what they're going to do is they're going to grab on, right? So up here at this exon, here's the intron, that's the intervening sequence. So this middle part, I want to get rid of it. So here this thing goes, oh, this is information we need. And this guy goes, hey, you know what, there's some more information we need down here. So they grab on to basically the mRNA. And what they do is these two guys right here, these four guys, these SNRAs and these SNRPs, they're going to basically loop together. And as they loop together, they make a lariat, a lasso, and they're going to cut that out. As they cut that out, some of these guys are going to act as enzymes, and they're going to then glue these two exons together. So there's going to be the intron that we've gotten rid of. There's going to be our mature mRNA. We still going to have to probably put our caps on it, and then we can send it out. But that's the spliceosome reaction to get rid of the introns. We don't need those guys in there. Now, what does this mean? Well, because we can basically cut these introns out and take these exons and move them around in different ways, that's how we can only have about 25,000 genes, but we have 100,000 different protein products. It depends on how you take these yellow pieces up here and put them back together. So in this first particular pattern, right, they took exons 1, 2, 3, and 4 and put them together. But in the second one, they took 1, 2, 3, 5, and 6. So depending on how you take these good pieces of DNA and put them together, this is going to give you a different recipe than this. This recipe is going to build some other type of protein than this. So from the same gene, you can get multiple different finished proteins. Okay? All right, so we're going to get into this as well. Here's our tRNA. Now remember, our tRNA, its whole job is to bring amino acids. Okay? So what's going to happen here? It's got an acceptor stem, that's the 3' end. This is going to be the end that's going to carry the amino acid. And down here, this is the anticodon loop. This is going to be a series of bases that are going to match up with your codons from your codon table. So start codon, AUG, well your anticodon would be UAC, and that's what would be there, and it would match up, and up here is where it would be holding methionine. Okay, so we have different models of this. Here you can see a little bit better the anticodon end because of the 3 spaces left. So what we have to do is we've got to take our tRNA and we've got to actually put an amino acid on it. I mean, if it doesn't have an amino acid, it's not really going to do a whole heck of a lot for us. So what we're going to do is we're going to go ahead and we're going to do this charging reaction. The tRNA, we're going to send it into this acceptor site. Okay, and this is a little enzyme called aminoacyl tRNA synthetase. So basically, it charges up the tRNA to carry an amino acid. So in this particular instance, in this first picture, you can see here's tryptophan. This is our amino group. Here's our carboxyl group. The side chain is going to be that little tryptophan thing right there. ATP is going to come in. It's going to drop off a couple of phosphates. As it does that, right, we've got adenosine monophosphate that's going to kind of hold on to this little accepting site as this tRNA molecule comes in. As the AMP leaves, what's going to happen is over here on this carboxylic acid functional group, this oxygen right here is going to get connected to our little acceptor end. And there's going to be a bond in there, and that's going to hold our tryptophan. So now this particular amino acid is going to hold tryptophan. I'm going to stop there for now. All right, I just wanted to get up to the charging reaction, go through some of the eukaryotic transcription. Be on the lookout, though, because I'm going to finish the translation stuff and make sure we understand how this goes through. But I wanted to get to the charging reaction, and all we're doing here is using some energy to create a bond between an amino group and our tRNA group, and we're using an enzyme to facilitate that. If you have any questions on any of this, do me a solid when I post this on Google Classroom. If you have questions, post them in the comments on Google Classroom, and that way I can address them. I'm thinking tomorrow maybe I'll throw out like a time for maybe an optional tutoring session, and if you guys want to pop into like a Google Meet, you have some questions, we can do a little face-to-face and get that squared away. So take a look at this at some point today, maybe post some comments. We can address that tomorrow maybe in like a little live stream meet. Alright, that's it. Have a great day, guys.